Good evening, Samesht. Okay, so today I'm Rahul Saha, and today we'll be talking about. Pratham had a query regarding this thing. I don't know where Pratham is. So he had sent me a message that he had a query regarding. Yeah. Okay, see today that corona is mutating and that yeah, it has mutated. A lot of mutations have occurred. We have this D614G mutation, aspartic acid to glycine mutation has occurred. One more mutation has occurred. Okay, it's a virus, and especially it's an RNA virus, right? It's an RNA virus, positive sense, non-segmented RNA virus. So RNA has number one uracil in the biomolecule thing here. In its second carbon, it has two. At this second carbon, it has OH group instead of only H. So that is more reactive to oxidation. Okay, that is more, you can say, vulnerable to oxidation, and thereby the repair pathway will not be able to recognize. Because why? Because cytosine, okay, deamination of cytosine directly results into uracil. One more thing is cytosine results into uracil. So if you look at the structure of uracil and cytosine, you will be able to make out. Okay, so I'll draw the structure for you. Let me draw it. So first of all, let's draw the structure of uracil and show you how and why does it mutate. Okay, this is N H N H double bond O, double bond O. This is nothing but uracil. Okay. Now once you talk about cytosine, its structure is like this. Here N. Just look how similar these two structures are. Both are pyrimidines, single ringed structures. O is NH2. Okay. Now, what you have is you have a double bond here, okay, and you have a double bond here. Now, what happens is you have oxidative deamination reactions happening due to UV rays, happening due to certain antibiotics, okay, that we take on regular basis. Okay, so in case of the plants, in viruses also, they are, you can say, susceptible to UV damage, okay, UV ray and all. So multiple damages they may have that may occur when they get into our body. Reactive oxygen species can cause that kind of damage. So deamination, if you take out this amine group, oxidative, cause oxidation, if you replace it with oxygen, what you get is uracil. Suppose in this strand of RNA, you had A, U, C, G, A. So this cytosine can be deaminated to, then suppose it should have been, the complementary should have been U, A, G, then C, then U. Okay, this should be the complementary. But suppose this has been deaminated to form uracil. Now the new sequence is A, U, U, G, A. But the repair pathway, the NSP14 present in the RNA virus SARS-CoV-2, non-structural protein 14, will not be able to detect it. Why? Because it is one of the common or intricate nucleotide reserve. So it is detecting uracil as its own. So now it's A, U, A, C, again U. Now its complementary is A, here it should have been G. So now this is a mismatch. And now in the later stage, when this will be carried forward, it will become mutation. There is difference between a mutation and a mismatch. Mismatch is this one. Okay. Parent is same. If you have parent is same, that is mismatch. But if parent and daughter both are having problem, okay. First of all, what will happen? Only uracil will come in. This will still be, you can say, guanine. But when both of these are different, that is known as mutation. And mismatch means one of this will be same, one will be having the deamination, that is known as mismatching. So this is how mutation is produced in RNA viruses quickly, okay, compared to any kind of DNA viruses, because DNA viruses have thymine. Thymine will have here H3C group, okay. This will then become thymine. So simple deamination of cytosine won't make it thymine, okay, can never make it thymine, okay. Good evening, Gayatri. Vaccine comes, we'll have to wear masks at least 2022. Yeah, good evening, Pratham. I was waiting for you. We need to go to school. I don't think so that you need to go to school. If you ask me, as a profound biologist, I would not recommend you to go to school whatsoever. Okay, I've read more than 1000 research papers on SARS-CoV-2. Okay, I don't know about this UP government. Okay, but the thing is, you never know. Gayatri also is correct only because you never know what is happening. Means what is your immune response? They speculate that only 2% are dying. But if you look at the people's death, okay, so I have my channel Rahul's Advanced Biology, you might have seen that. So in that I had put 2-3 videos of the drugs being used against SARS-CoV-2. One of them was tocilizumab, okay. So the tocilizumab, narsoplimab, these are the monoclonal antibodies against, you can say, complement pathway and interleukin 6. So one of the many patients have reverted to me on my channel saying that I lost my brother who is 31 years of age. I lost my sister who is 28 years of age. And why did he not recover after whatever drug I had, you can say, made a video on? That same drug was administered to them by the doctor. Okay, the thing is, you do not know what is the immune response. You have D-dimer formation, clotting pathway, then kallikrein kinin pathway, interleukin pathway, interleukin 1, 6, then you have complement inhibitory pathway, okay, you have anaphylotoxins, so many things are there. You have the, you can say, MASP pathway, okay. So many pathways are there in immunology that are being targeted by this SARS-CoV-2. It also hijacks our translation system, which makes the proteins, okay. It also hijacks the type 1 interferon system. So one research paper yesterday it came out that if you have deficiency in type 1 interferon, then you will be more vulnerable to the SARS-CoV-2 virus, and then you can produce ARDS, that is acute respiratory distress syndrome. Okay, you have fluid formation or the fluid accumulation, that is the pleural fluid in the lungs, okay, that gets accumulated and then we eventually die. And then we have sepsis, we have multiple organ failure. Even there was a research paper that showed that if SARS-CoV-2 crosses the blood-brain barrier, then even if we recover, our brains would be 10 years older, okay, 10 years older to what our brain is now currently. So that's a huge, daunting, you can say, tumultuous task that we are putting ourselves through, okay. So one should not go to school. You can write it down, do not give consent, okay. So Gayatri, the thing is fine, but next you can go, okay, after vaccine is administered you can go, right? Yeah, after effects, that is what I was telling you. Will have, you can say, the after effects are so deadly that 10 years of cognitive decline you will be having, okay. You can have diabetes onset also after effects, okay. People, even the head of the NIH, National Institute of Health, NIAID, you can say the director of that, Anthony M. Fauci, he has also speculated that the vaccines will come, but since they are accelerated vaccines, even if you talk about Pfizer and BioNTech, you talk about this AstraZeneca, you talk about, you can say, Moderna's mRNA 1273 vaccine, all of this, they may have some side effects because they're accelerated vaccines. Vaccine development take years and years of time. A lot of, you can say, clinical trials should be done. Huge, large scale clinical trials that cannot be accelerated. But the times are as such that they had to be accelerated. Okay. Sputnik 5, do not put it in your body, Gayatri, at all if it comes in India. Sputnik 5 is a type of vaccine. I'll send the research paper to you. Okay, the research paper was published in a very quality journal. Okay, impact factor of 70, known as New England Journal of, you can say, Medicine, NEJM. It showed that it makes you form antibodies. Okay, but they are not really neutralizing antibody. What they are is, they are non-effective antibody. Those antibodies cannot kill SARS-CoV-2. So what's the use of such antibody? Okay, so Russia just wanted the share. It wanted a great capital from that vaccine that people will just buy due to the hype. Okay, the best vaccine as per me is going to be either Moderna's one, okay, Moderna mRNA 1273, this vaccine, or Pfizer BioNTech, okay, BNT 1262, okay, BioNTech vaccine. So these two AstraZeneca's, you can say, AZD 1222 vaccine is a good one. Only problem is that it is using adenoviral vector 5. So adenovirus can sometimes cause side effect. Okay, side effect in the sense it can lead to immune reaction. Immune system can recognize it. Okay, it can lead to deadly side effects also sometimes. Even though it's a disarmed adenovirus, you won't be knowing what adenovirus is. We have multiple ways to produce vaccine. Okay, we can produce it via lentivirus. Adenovirus is the most effective one. Okay, that's the only problem with Oxford's vaccine that I feel. Okay, even though it is disarmed adenovirus, it should not be causing such problems, but that's the greatest problem that we can think of. Okay, that is, so be safe, that's all. Okay, you need to wear masks and don't go to school. Online classes are the way to go, at least till the end of this session, till March. Okay, from April you can think of, because by that time you may be getting at least one sort of vaccine. Okay, so one sort of vaccine will provide you with, you can say, enough protection, immunity, cell mediated, against the disease. Okay, so Pratham, you had that query regarding the muscle proteins, right? So you want me to explain the actinin and all, actinin and nebulin and all this. Yeah, that is, they want money. What do they want? That is all money minded creatures, that's all, nothing else. Okay, okay, Pratham, so I'll explain it to you. So I'll just revise the zones for you, okay, the discs and the zones, so that it remains in your head. So what is a Z disc? Okay, Z disc is, okay, if you are attempting test tomorrow, then Z disc, okay, if you are talking about the Z disc, Z disc is nothing but it is a narrow plate shaped region of dense material that separates one sarcomere from the next. That separates one sarcomere, okay, from the next. That is our Z disc. Okay, then A band. This should be in your head all the time, very, very precisely. Okay, these concepts are very, very important. They are dark middle part of sarcomere that extends the entire length of thick filament and includes some parts of thin filaments that overlap thick filament. So made up of, it is known as thick filament, made up of mainly myosin fibers. Okay, now coming to our special I band. Okay, so when we talk about I band, they are the lighter, less dense, thin filament. Okay, they contain only thin filament, no thick filament. Okay, thin filament. Okay, actin fibers. Okay, F actins. Okay, and a Z disc passes through the center of each I band. Always remember, Z disc passes through the center of each I band. Very important NEET question. A Z disc passes through the center of each I band. Okay. Then next up we come to H zone. H zone is what? It's a narrow region in the center of a band. A narrow region in the center of a band. Okay. That contains thick filaments but no thin filaments. Like I band does not contain, you can say, thick filament. This contains no thin filament. That contains only thick filaments. Only thick filaments. No overlapping. Okay, no overlapping happens in this H zone, because A zone and towards the end the overlapping will happen. At H zone no overlapping will happen. I'll just show you the image also. It will be very, very clear. Okay, coming to the M line. So M line is nothing but it is the region in center of H zone. Okay, it is the region at now the center of H zone. Center of H zone. Okay, center of A is H. Now center of H is M line that hold thick filaments together at the center of sarcomere. It holds the thick filaments together at the center of sarcomere. Okay. So let's see if I can find an image for you. Zones in muscle fiber. Yeah, you'll get good images like this. You'll just see this image. It's a good image. Okay, just see A band. You have little bit of, you can say, overlapping. H zone, only the thick. Okay, then you will be having, you can say, M line. At the center of H is the M line. Okay, here that is the M line. Center of this is the M line. Is I band. Okay, Z is at the center of I. You can see Z is at the center of I. Always remember these things. Thin are actin, thick are myosin. Okay, so this is how the bands are located. Okay, so first of all, you must be acquainted and always know the reason and the function behind each and every band. Then we will come to the proteins. Okay, now we'll come to the muscle proteins. Very, very important from your NEET perspective. Okay, muscle proteins. So I'll just tell you the functions in short. Myosin actin we all know. Okay, so now tropomyosin. Now tropomyosin binds where? To the actin binding site. Sorry, myosin binding site on actin. It binds where? It binds to the myosin binding site on specialized G-actin polymer, or simply you can call it F-actin also, because F-actin is a polymer of G-actin. So if it is actin is like this, multiple G-actins are associated. These are G-actins. Okay, G-actin, and this entire sheet is F-actin polymer. So each one will have a specific site, okay, which binds to the myosin. So that will be covered by tropomyosin. That is covered truly by tropomyosin. And troponin, I had told you that troponin is attached to tropomyosin and has three specific subunits: troponin I, troponin T, troponin C. Okay, so troponin C part, it binds to the calcium ions. Now calcium, how does it come in? That I had described to you already, because when you have the, you can say, to the acetylcholine receptors, acetylcholine binds, okay, then what will happen is it will cause the sodium ion to come in through that part, and then what will happen is the signaling will continue, and then that will lead to the release of calcium from the sarcoplasmic reticulum, which has a lot of calcium via the ryanodine receptors. Okay, the action potential will go through and reach the T tubule, reach the triad, T tubule, and the terminal cistern of the sarcoplasmic reticulum, if you remember in the last class, and then that will lead to the opening of the ryanodine receptor, or simply SR receptors, which will take out, or which will start throwing out Ca2 plus. And Ca2 plus, when it binds to the troponin C residue, will cause the troponin I to have conformational change, which will pull, which is then attached to the tropomyosin, which will pull tropomyosin and make the entire this thing. Troponin I will be attached, so T is there, C is there, C will attach to calcium ion then, which is released from the sarcoplasmic reticulum. Okay, and then it will pull, and then this will also get pulled, and then this will be free. These, you can say, subunits will be completely free, and then, or the binding sites or the active sites will be completely free, and then actin, the myosin with its myosin binding head, which has got the actin binding site in that, you can say, will be able to bind, because one of the heads will have ATP binding site, one of the heads will have the actin binding site. Okay, so it will bind to the myosin binding site of actin. The rest of the proteins, when we come on to, okay, one protein is titin, 3 million Dalton. I told you the entire weight of titin, one of the largest proteins. Okay, this is the structural protein. I'll write the function again of titin. This is the structural protein. Okay, that connects the Z disc to M line of sarcomere. Very, very important. Connects Z disc to M line of sarcomere. Very, very important. Okay, so what it does is it helps to stabilize the position of the thick filament itself. Okay, and can stretch and then spring back, you can say, undistorted. Okay, and thus it accounts for much of the elasticity and also the extensibility of the myofilaments. Otherwise the myofilaments can disassociate from each other. Okay, so when the Z disc is connected to the M line, okay, now you know what is M line already. Okay, M line is at the center of the H zone. Okay, and Z disc is the narrow plate which is present at the center of the I band. So Z disc and M line, when they are connected, means what? And S zone is what? S zone is at the A band, means A band and I band are tightly associated. Their entire sarcomere between 1Z to 2Z. Okay, that is the entire sarcomere from 1Z to 2Z. So the entire sarcomere is then stabilized. Okay, its position is not broken, or its position is not, you can say, destabilized. That is the work of titin. Now, once we go to the alpha actinin, okay, alpha actinin. So in case of alpha actinin, the structural proteins which are present at the Z discs are alpha actinin that attaches to actin molecules of the thin filament, because Z disc is what? It is present at the center of the thin filament, so it must have some protein for it to attach to the thin filament. Thin filament is what? Made up of actin only, right? So the Z disc comprises of alpha actinin. Alpha actinin is the structural protein of Z disc. Directly you can say structural protein pertaining to Z disc, which will attach itself to the thin filament or actin, because Z disc is present at the center of the thin filament. Okay, and also it attaches itself to the titin molecules also. One more function is it attaches itself to titin molecules also, or to titin protein. Attaches to both the actin protein and the titin protein. That is the work of Z disc, because Z disc is what? I told you titin is the structural protein that connects Z disc to M line. Okay, now Z disc is made up of what? Again alpha-actinin. Now see the concept here. So this is connected to here. Z disc connected to M line means Z disc contains what? It contains the main structural protein is the actin. So actin will be connected to titin also. Okay, if you understand, this is how the concepts, they are all interlinked. Okay, now coming to the third protein known as myomycin. When we talk about myomycin, that is the structural protein that forms the M line of the sarcomere. Very, very important. Okay, forms the M line of the sarcomere. M line is the center of the H zone. H zone is the center of the A band. So M line is produced by myomycin. Okay, forms the M line. Okay, structural protein that pertains to the M line of sarcomere, and then M line will do what? M line will again bind to titin, because titin is what? It binds to the M line of sarcomere and Z disc. So titin is binding to, you can say, alpha-actinin also, and it is binding to myomycin also. Again, interrelated concept. It binds to titin, and titin is now binding to alpha-actinin of Z disc and myomycin of M line. Okay, again one more interrelated concept. So it is, they are all interrelated concepts, right? So that is the work of myomycin. So it binds to titin molecules and connects the adjacent thick filaments to one another. Okay, function is connecting adjacent thick filaments to one another. Okay, that is the work of myomycin. Now we come to the work of nebulin, or special protein known as nebulin. When we talk about nebulin, it is the structural protein, very, very important, a very significant protein. It is the structural protein, okay, that wraps around the entire length of thin filament, means actin, that wraps around the entire length of, you can say, thin filament, that is actin. Slash actin protein. Okay, and this will do what? Okay, this in the end helps anchor the thin filaments to the Z disc, because Z disc is binding, Z disc is having alpha-actinin, it is binding to actin also, and then it will bind to nebulin also. So Z disc will be able to hold on to the thin filament in a better way, more efficiently, more tightly. So helps anchor Z disc, okay, helps it to anchor Z disc. Okay, and then what will be the application? It will regulate the length of thin filaments during the development of muscle fiber. Because when we exercise, we do what? We go for hypertrophy, right? If you have a huge bicep, means you have hypertrophied the muscle, meaning that you have increased the length of the thick and thin filament, mostly the thin filaments. Their lengths have increased, okay, or their numbers have also increased. You can increase the number also, okay. That is the meaning of hypertrophy of muscle fiber, okay. Increasing the size of the myocyte, okay, increasing the size of the muscle cell slash muscle fiber slash myocyte. So regulate the length of the thin filament during development. Okay. Then we come to our dystrophin. I should say this is the most important protein. It is overlooked while teaching, but it is the most important protein. That's why I have included this one also. DMD question is asked in NEET also. Duchenne muscular dystrophy. Okay. So dystrophin. It is such an important protein that it helps to link the thin filament of sarcomere to the integral membrane proteins present in the plasma membrane of sarcolemma. Thin filament of sarcomere is attached to the integral membrane protein of or present in the sarcolemma. That is the work of the sole purpose of your dystrophin protein, okay, which are in turn attached to proteins in connective tissue matrix. Means the sarcolemma, the integral membrane proteins of the sarcolemma are in turn attached to the proteins in connective tissue matrix, that is the extracellular matrix which surrounds the muscle fibers, and this reinforces, you can say, the sarcolemma and helps transmit the tension generated by sarcomeres to the tendons. Because I told you that epimysium, perimysium, endomysium surrounds the tendon. So how will the length tension relationship cause the movement of the bones? Because tendons, it connects the muscles to bones. How I am able to move this hand? Because my bicep muscle is able to transmit the bicep and the tricep both is able to transmit the tension or the force. Tension means force in Newton generated by the sliding filament theory, the contraction to the bones. So if I cannot transmit that force to the tendon, can I move my hand? Can I move my limb? No, I cannot move the radius and ulna bones. I cannot, because the tension has not been transmitted or it has not traversed and reached the tendon proportion. The thing is, in order for it to reach the tendon, you should communicate cell to cell. If muscle contraction is happening, but it is not being able to reach the cell membrane only, the sarcolemma only, how can you think of the cell shortening, the muscle shortening? So the muscle cell will shorten and that will transmit the entire force to the extracellular matrix and thereby towards the tendon. If the muscle cell cannot shorten, only the actin and myosin they shorten, but the muscle cell as a whole cannot shorten, then it cannot be transmitted. The force or the tension cannot be transmitted and thereby you will feel like you are paralyzed. It's not a nerve disease, but still you cannot move a limb. So one of the worst kinds of dystrophy is Duchenne muscular dystrophy, which happens due to a specific missense mutation in the dystrophin gene coding for the dystrophin protein. Right? So gene therapy is the only cure. People have progressed a lot. The scientists have made a lot of progress when it comes to, you can say, dystrophin, you can say protein and its gene therapy. Okay, so scientists have made a lot of breakthrough. Okay, don't worry, but scientists have made a lot of breakthrough in it. Okay, so now we'll proceed to the types of joints. Okay. Yeah, I'll surely take up the sliding filament theory. Okay, don't worry, Gayatri. I'll surely take up the sliding filament theory. Okay, that is there. So that will be taken. Okay. So now we'll come to the classification of joints. Okay. So joint classification. Okay. So joints are classified structurally based on their anatomical characteristics. Okay. And functionality also. So based on the type of movement they permit. So the structural classification of joints is based on two criteria. Number one is the presence or absence. So if we talk about joint classification, so the structural classification of joint is based on two criteria only. Okay, the presence or absence of a space between the articulating bone or connecting bone. Okay, articulating bone is the connecting bone. The presence or absence of an articulating or presence or absence of space, I should be saying. Okay, the presence or absence of space between an articulating bone, that is the connecting bone, okay, which is known as the synovial cavity. I'll just show it to you what is a synovial cavity, because you have synovial joints in your syllabus, known as the synovial cavity. The next point is the type of connective tissue that binds the bones together. Okay, the type of connective tissue that binds, you can say, the bones together. These are the two criteria. I'll just show you the synovial cavity to you. Okay. This is known as a synovial cavity. Just I'll zoom it in, go to Wikipedia. Look at this image. Okay, this is the articulating cartilage, means the joining cartilage. This is the synovial membrane. Okay, this is the entire articulating capsule. Okay, and this is the synovial fluid or the synovial cavity. So this is the fluid which doesn't let the cartilages come into contact when you move your knee. And when the synovial fluid production is reduced, because this is the synovial membrane, this one is responsible for production of synovial fluid. So when you, if you take immunology or you take orthopedics, then you will be learning how does the synovial membrane produce synovial fluid. So most of the joints in our body which can rotate, okay, freely, you can say ball and socket, hinge joint, saddle joint, they are all synovial joints. Like your shoulder joints, the elbow joint, the knee joint, the ankle joint, they are all synovial joints. Okay, so the thing is, the synovial joints can easily, what it can do is, they can, synovial membrane can produce, you can say, synovial fluid, and this fluid acts mainly as a shock absorber. It has got immunological properties also. It has got monocytes and macrophages. So then when it has got too many inflammatory markers like IL-6 and IL-1, tumor necrosis factor alpha, then what happens is that can cause degradation of the synovial membrane, okay, and then synovial fluid production is reduced as we age also. And then what happens is, with every knee movement these cartilages will collide, and when these cartilages will collide, what will happen is, in the end, what will happen is, you will be facing, you can say, what will happen is, you will be facing something like cartilage disintegration. This cartilage will get disintegrated completely, and when this cartilage gets disintegrated completely, what happens is, in the end, the bone to bone collision will start happening. The bone to bone collision will start happening, and when bone to bone collision starts happening, then what you see is, you have something known as osteoarthritis. Okay, you have something known as osteoarthritis. That is the problem that we face, okay, when we get old. Osteoarthritis problem. So if you have this synovial cavity and the synovial fluid is being produced properly, then your joints will be completely fine and you will not have any kind of problem or ache in the synovial joints. So based on that classification, okay, what you have is, you have something known as, three types of joints are present in our body. Number one is the fibrous joint, okay. If you come to types of joints, okay, you might have read of, you can say, saddle joint and all, those come under the synovial joint, but typically we have three types of joints. Number one is the fibrous joint. Okay. Number one is the fibrous joint. So in a fibrous joint, there is no synovial cavity, okay. There is no synovial cavity in the fibrous joint, always remember. So good neat question also. There is absence of synovial cavity, and bones are held together by dense irregular connective tissue. Bones are held together by dense irregular connective tissue. Okay, this dense irregular connective tissue is rich in triple helical collagen material. Is rich in collagen. It's rich in collagen fibers. So it is extremely reinforced, you can say. Okay. Yeah, rheumatic arthritis, Gayatri. This is the same thing that I was talking about. Osteoarthritis, rheumatic arthritis, they are all various types of arthritis. Arthritis means articulating joints. These joints have inflammation. Inflammation means itis. Itis is the suffix. Suffix means inflammation. Inflammation means the dysfunction of any biochemical pathway. So sometimes what happens is, you have something known as, you can say, inflammatory attack. Okay. So there are certain individuals who are HLA-B27 positive. Okay, HLA is human leukocyte antigen. Okay, so HLA-B27 is a type of HLA. If you are positive for it, you are more prone to developing inflammation in the joint. Inflammation means you will be having more production of extremely inflammatory molecules like tumor necrosis factor alpha, interleukin 6, interleukin 1, okay, interleukin 17, okay, so then interferons, okay, interferon gamma. So there are many types of proteins which are upregulated. Okay, these proteins, what they do, they completely break down the synovial membrane. Okay, and then they can attack on the cartilage also. They can break down the cartilage also, and the cartilage will get abrasive as the time goes by, and then it will completely destroy the cartilage. It will prevent the formation of, you can say, the synovial fluid, and in the end, bone to bone collision will happen. You have to go for knee replacement therapy. So the drugs available are monoclonal antibodies which block these molecules. You have IL-6 inhibitors. One of the videos I had made on my channel is tocilizumab. So tocilizumab is an anti-rheumatic drug that you are asking. IL-1, anakinra. There is a drug known as anakinra. IL-1, you can say receptor antagonist. IL-6 receptor antagonist means wherever IL-6 will bind, okay, it will cover that specific receptor, so IL-6 cannot cause any kind of damaging effect. Okay, so there are many chemicals which are upregulated in the human body, okay, because of HLA-B27. Even B27 negative people can have these chemicals. Again, the question to answer to that question, the research is going on that why all of a sudden these chemicals are activated. One theory is why these chemicals are too much high in some people is gut dysbiosis theory, meaning that you have 10 to the power 13 to 10 to the power 18 cells in the colon, okay, in the colon. That is known as cells of bacteria, fungi, viruses. Also you have methanogens also, streptococcus, you can say thermophilus, you can have a thermophile also. So these archaebacteria are also present, so they are a great exemplar of co-evolution, okay. So most of these bacteria are, or these microorganisms are favorable microorganisms, okay. So their metabolites, okay, can also reach the brain. So last year there was a great research revealing the gut-brain axis. Gut-brain axis. So the brain problems, okay, the implications of brain problems like, or the brain neurodegenerative ailments like Parkinson's, you might have heard of, where you have tremor, like this tremor in the hand, you cannot write, you will be having tremors. You will be having Alzheimer's, the worst form of dementia, where the neurons will start to die, known as degeneration, okay, and you will be having memory loss, and in the end you will die. In Parkinson's also, there are dopamine secreting neurons. If you have heard of dopamine as a neurotransmitter, so in the midbrain there is a portion known as substantia nigra. So there in that region, dopaminergic neurons, they degenerate or die. So because of these specific metabolites, if there are too many harmful bacteria, then harmful metabolites can be generated. They can reach the brain and they can cause that disease. Same way, harmful bacterial metabolites can reach the various joints, or even the harmful bacteria can reach various joints, and then our immune system will do what? Will try to clear that, and when that bacteria will mount an offense, immune system will try to clear it. Immune system will generate these IL-6, IL-1 chemicals, and in turn this IL-6, IL-1 will have or will cause collateral damage, because they'll try to reduce inflammation, clear out the bacteria. Macrophages will come in, macrophages will be activated, monocytes will be activated to form macrophages. Then also there are two types of macrophages, M1 and M2, okay. What will happen is you will have collateral damage, like macrophages are throwing out chemicals known as cytokines released from cells. So these cytokines are only these IL-6 and IL-1 and TNF alpha and all. So these will try to, you can say, nullify the bacteria, but collateral damage is it will destroy the surrounding tissue also. So that is one reason behind excessively high level of, you can say, these specific chemicals in the people who suffer from osteoarthritis. So it all stems from the gut, okay. So if our bacteria is in proper concentration, we do not have too much bad bacteria, we have proper bacteria, then it's not a problem. But once we have too much of harmful bacteria, a condition known as dysbiosis, means it is out of, dys means functional, biosis means the bacteria culture or the bacterial cells are not what they should be. Harmful bacteria are becoming more and more, useful bacteria are becoming lesser and lesser. So that tends to increase the hyperimmune response, and collateral damage is tissue damage. Like if I drop a bomb there, and if one terrorist is there, I'll drop a bomb. So the bomb will kill 10 more innocent people who are standing nearby. So the terrorist is killed, but the nearby people are also killed. So that is like collateral damage, Gayatri. Okay, that is how it happens. So that is how we can think of what is happening. Okay, so the next type of joints that are there, okay, are cartilaginous joints and synovial joints. Okay, so cartilaginous joints Joint number 2 is cartilaginous joints. Okay, in cartilaginous joints, there is no synovial cavity again, and the bones are held by cartilage. No synovial cavity. The bones are held by cartilage. Okay. Number 3, you have synovial joints. You have a synovial cavity, just like I had showed you. Synovial joints. You have a proper synovial cavity, proper synovial fluid. This one which I had shown you. Okay. So the bones forming the joint have a synovial cavity, you can say, and are united by the dense irregular connective tissue of an articular capsule, you can say, which is like an accessory ligament, the ligaments that I had shown you, okay, in the knee. So those will be the articulating ligaments. So this is the entire articulating capsule. Under that you have the synovial membrane that produces the synovial fluid. Okay, gives great mobility. So synovial joints have maximum mobility. Cartilaginous joints are little bit less, and fibrous joints are not mobile. Just like the bone of my skull, there are 8 bones. Can you move any bone separately? No. These are all fibrous joints. So we'll talk about the examples and all in detail more in the next class. Now I think your test is there. So go for your test. If you have any kind of doubts or queries, you can contact me on my personal number so that we can discuss more about your preferred topics in the doubt class. Okay then.